So, good afternoon to the biodiversity community. My name is Selly Ngaloumo Abdel Aziz, and I'll be giving a short talk on evolution of genetic diversity of Plasmodium falciparum in Cameroon. I will be presenting as outlined. Over the long course of human history, the infectious disease that has killed more humans than any other is malaria. Malaria is caused by a protozoan of the genus Plasmodium, which is carried and transmitted through the bite of infected female Anopheline mosquitoes. In 2020, there was an estimated 241 million malaria cases and 627,000 malaria deaths worldwide. Sub-Saharan African countries carry a disproportionate share of the global malaria burden, that is 95% of malaria cases and 96% of malaria deaths. Malaria was estimated in 2020 to kill approximately 10 persons each week in Cameroon. This makes no sense given that we have known how to cure malaria since the 1600s. That is when the French scientists discovered the back of the cinchona tree, which contains quinine, which is still an active malaria treatment till this day. In the 1950s, there was a lot of optimism about the idea that malaria might be eradicated. And all this optimism stemmed from the fact that wonderful drugs and insecticides were present at that time. At that time, chloroquine had been discovered to be a highly effective anti-malaria drug, which was safe, cheap, and affordable, and DDT approved to be a highly effective insecticide against mosquitoes. But in the 1990s, there was a glowing disappointment, since it seemed impossible to achieve global malaria eradication. And this was due to a huge financial requirements and a complex state-of-the-art logistics which needed to be set up. But the crucial factor was now that the parasite had become resistant to frontline anti-malaria drugs in circulation. In order to cure this burden, WHO set a certain numbers of strategies to be able to eradicate the burden, which was the use of chemoprophylaxis, the use of vector controls, and now we have the use of vaccines that have been proposed. Despite all the effort and measures put in place, the malaria epidemic still strives. When the malaria parasite is attacked by the immune system and by drugs in the human body, its genetic and evolutionary defense mechanisms come into play, activating the process of resistance mechanism. And crucial question is, how often do malaria develop resistance or develop mutations in their genome? And the answer here will be that the malaria parasite will generate a new mutation after every 4 to 5 positions of the parasite genome, which is a 23 million base long, roughly every couple of days in a single infected person. That is giving rise to millions of polymorphisms that can be found within the malaria parasite gene. As a result, the parasite genome is constantly shuffled, resulting in antigenic diversity in the forms of millions of new variants every couple of days. Even though the predominant cause of parasite virulence is genetic variation, that is genetic diversity, genetic variation still then will lead to natural selection, which is necessary for evolution. Natural selection is said to be slowed in areas with gene flow, with high gene flow, and high malaria ecological areas like Yaounde that experiences high gene flow due to the large population movement in and out of the city may then be a region with high Plasmodium falciparum genetic diversity. As a result, the primary goal of this research was to study the influence of massive and intense population movement on the evolution of Plasmodium falciparum genetic diversity in Yaounde, Cameroon, using the Random Amplified Polymorphic DNA PCR technique and also the nested-PCR technique where we targeted the MSP2 genes. The study was conducted in Yaounde population for 2 time points, that is in 2024, that is 2014, that is before, and in 2018, that is during the intensive massive movement of people in and out of the city. With this, we came out with certain results, such as our results showed no significant difference in the multiplicity of infection in 2014 and in 2018. When we know that multiplicity of infection is a parameter used to study the transmission levels of the parasite. Hence, from the multiplicity of infection, we could confirm that the fact that Yaounde is a high transmission region. The calculated mean genetic diversity parameters across years, that is the Shannon index, multiplicity of infection, and the naive genetic diversity, show that the parasite population in Yaounde are highly divergent. Clustering analysis was also used as a proxy for population structure, with the MSP2-based UPGMA distance depicting intermixed and epidemiological related cloned populations of 2014 and 2018 as shown on the graph above. Interestingly, the distance tree based on RAPD primers revealed 2 clear, distinct, unrelated clusters from 2014 alone, suggesting different genetic backgrounds from the rest of the intermixed populations. To conclude, we will say these observations warrant in-depth investigations into how human migration redistribute malaria diversity and the interest of targeting populations with high genetic backgrounds for genetic diversity purpose. Thank you for listening.